Once again, welcome back with another microbiology lecture, sterilization. In this video lecture, I am going to teach you sterilization, some terms that are related to sterilization and types of sterilization. Let's start the lecture. So what is sterilization? Sterilization is the complete destruction of all living cells, spores and acellular entities. For example, the living cells include bacterial and fungal cells, and acellular entities include viruses, prions or virides. Sterilization is an absolute term, that is, a sterile object is totally microbes free, that is, from bacterial, fungal spores and infectious agents or particles. For example, viruses, prions, virides. Sterilization is achieved by killing or removing microbes. Then we have antimicrobial agents. These are chemical agents that kill or inhibit the growth of microbes. As we know, sterilization is the complete destruction of all form of microbes and spores, while disinfection does not cause complete destruction because vegetative cells are killed by disinfections while spores are not killed. That's why disinfection does not cause complete destruction. Microbial colonies or number are reduced in disinfection to a state where they are not harmful to health. Disinfections include formaldehyde, glutaraldehyde, hydrogen peroxide and iodophores. Then we have another term, disinfectants. These are antimicrobial chemicals that are used on non-living surfaces. They are not harmful to living tissues. The example of disinfectants are alcohol, bleach. Then we have antiseptics. Antiseptics stops or slow down the growth of microbes. They are frequently used in hospitals and other medical settings to reduce the risk of infections during surgery and other procedures. They are safely applied on living tissues, used in wounds, cuts, etc., and can kill or inhibit microbial growth. The example of antiseptics are chlorhexidine, hydrogen peroxide, pyodine, iodine, etc. Then we have purpose of sterilization. It means that why sterilization is necessary. It is necessary to make objects microbes free, to achieve complete destruction of microbes, to sterilize surgical equipments like dissecting forceps, tissue clamps, surgical probe, etc., etc., and to reduce risk of transmission of microbes to healthy person, to make equipments ready for use without harm, and also for health care professionals and patient safety. Then we have methods of sterilization. We have three methods of sterilization, that is physical method, chemical method and mechanical method. In this video lecture, we will discuss physical and mechanical method. Chemical methods of sterilization will be discussed in part 2. Physical method include dry heat sterilization, moist heat sterilization and sterilization by radiation. Let's discuss each in detail. In physical method of sterilization, dry heat can be used to sterilized item. Dry heat destroys microbes by causing denaturation of protein. They include natural method, flaming, hot air oven, incineration and pasteurization. Natural method of sterilization. As we know, sunlight have an active germicide effect due to presence of ultraviolet rays. It is natural procedure for sterilization which reduce number of microbes in water tank and lakes. Then we have flaming. Flaming is the simplest method for dry heat sterilization, in which material are kept at hot part of Bunsen burner flame or for few seconds until become red hot. For example, forceps, wire loops, etc. Then we have hot air oven. It consists of metallic chamber of aluminium and stainless steel, which is electrically heated and thermostatically controlled. They have two types, that is gravity convection type, in which air is circulated by gravity convection, and mechanical convection type, in which air is circulated by fans. They have heat capacity 50 to 300 degrees centigrade, but we usually set it 160 degrees centigrade for 1 hour. It is used for glasswares, conical flasks, culture plates and tube, etc. Next we have incineration. It is waste treatment process in which heat is used to sanitize medical or other hazardous waste before it is discarded with non-hazardous waste. It is basically destruction of waste by burn. It is used for pathogenic materials and hospital wastes. Now we have advantages and disadvantages of physical method of sterilization. They are suitable for dry substances, suitable for substances that destroyed by moisture, and glasswares like flask, test tubes and pipette can be sterilized through dry heat sterilization or physical method of sterilization. The disadvantage of physical method of sterilization: they cannot use for volatile and thermolabile objects. Long heating time and temperature is required for this process. Also not suitable for surgical dressings or plastic substances. Physical method of sterilization include moist heat sterilization, in which sterilization is achieved by boiling, autoclaving and pasteurization. Moist heat kills the organism by denaturing their enzyme and structural proteins. Most resistant spores generally require 121 degrees centigrade temperature for 15 to 30 minutes. It is used for sterilization of culture media and all other material through which steam can penetrate. They include boiling. Let's talk what is boiling sterilization. The instrument are boiled at 100 degrees centigrade in a clean container or sterilizer at least for 1 hour, so the germs are totally destroyed. Boiling method is useful for blades, knives and scissors. They are completely dipped in water. Next we have autoclave. Autoclave is a modified pressure cooker constructed for sterilization for surgical instrument, in which 121 degrees Celsius for 30 minutes. Autoclave is used for sterilization by steam under pressure. This saturated steam under pressure is allowed to penetrate through the material for 20 minutes at 121 degrees centigrade. Pasteurization is treatment of food items in a temperature by which mesophilic microbes are killed. Pasteurization destroyed most of microorganisms that cause food spoilage and so prolong the storage time of foods. It is mainly used in dairy products, that is packed and unpacked food items. There are two types of pasteurization. Number one, high temperature short time, and number two, low temperature long time. High temperature short time, also called flash method, in which 71.6 degrees centigrade temperature for 15 seconds is given to food items, and low temperature long time, also called batch pasteurization, in which 62.8 degrees centigrade temperature for 30 minutes is given to food items to achieve sterilization. Then we have radiation sterilization, which also known as cold sterilization, because there is no heat in this method and the microbes are destroyed by lethal effects of hazardous radiation. They include ionizing radiation and non-ionizing radiation. The ionizing radiation include gamma rays, X-rays, cosmic rays, which are used for culture swabs, urine catheter, drip sets, and all plastic objects, while non-ionizing radiation include ultraviolet rays and infrared rays, which is used for sealed item, for example ampoules, syringes, needles, urine bottles, labs and OTs, etc. Next we have mechanical method for sterilization, which include filtration and preservation. Filtration involved in the physical removal of all microbial cells from liquid and gas which are denatured by heat. Filtration is used for antibiotics, amino acid, vitamins, or injectable drugs most commonly. Then we have preservation. Preservatives is a process in which any food item used to keep in its original forms. Preservatives are non-toxic. The examples of preservative are nitrates, sulfur dioxide. Actually, preservatives are static agents used to inhibit the growth of microorganisms, most often in food. If eaten, they are non-toxic. Chemical sterilization will be discussed in part 2. Thanks for watching. Don't forget to subscribe.